morning everyone. My topic for the seminar is radioimmunoassay. In this session, we will be discussing about what basically radioimmunoassay is and the principle on which it works. And again, we will be discussing about the applications of radioimmunoassay. So, to start with, what actually radioimmunoassay is. As evident by the name radioimmunoassay, it is basically a test that is used to determine the concentration of antigen present in blood sample in patient sample. So, why is it named as? Why is it specifically named radio? Because we use radioactivity technique to measure the concentration of antigen present in patient sample. So, how does it actually work? So, now that we know what radioimmunoassay is, let's now move on to the principle of radioimmunoassay. The basic crux is that there are two types of antigens that bind for that compete for the binding sites on the antibodies, for a fixed number of antibodies. So there are two types of antigens that compete for a limited number of antigen binding sites on antibodies. The two types of antigen that compete are hot antigen and cold antigen. Hot antigen, why is it called as hot antigen? Because it is attached to radioactive element like iodine, carbon C40, and this tritium, carbon isotope of carbon, and this isotope of iodine. So, hot antigen is basically an antigen preparation that is coated with these elements. Those are radioactive elements. And the cold antigen is the unlabeled antigen that is obtained from sample. From the cycle of from the patient sample. So what happens is they both compete for the binding sites on the antibodies. So the binding of cold antigen to the antibodies present on the microtiter plate. So this is the basic principle. The cold antigen competes with the hot antigen. So in the next process, we'll learn how actually competes and how we'll actually determine the concentration of antigen present in the sample. So let's now talk about the steps involved or the process, how actually it happens. First, we take antigens, hot e.g. incubated into pre-coated microplate. This is the first step. Second step, hot e.g. binds to antibodies in microtiter plate. This is what a microtiter plate actually looks like. These are actually wells that are coated with antibodies. So selected number of antibodies, selected antibodies are used. So after this, what happens after this? Hot antigen binds to the antigen present in the microtiter plate. Antibody present in the microtiter plate. So after this, washing is done. Washing with suitable buffer. So that the unbound hot antigen is washed away, and then cold antigen is introduced. What this cold antigen actually does is it binds to it replaces the hot antigens that are bound to the antibodies in the microtiter plate, and the cold antigens they replace them, and the amount of the actually replaces the hot antigen is what forms the flux, and it is where it forms the basis from where we actually calculate the amount of the concentration of cold antigen. That is the patient, that is the antigen present in the patient sample. So to explain it in proper way, these are antigens, selected number of antibodies. In first step we had the hot antigen, they go and bind to antibodies, and then washed off. Few unbound antigens, hot antigens are washed off, and then introduction of cold antigens. Few of them cold antigens replace the hot antigens, and how actually the concentration is determined, that we discussed about. The amount of hot antigens displaced from the solution by cold antigen is inversely proportional to radioactivity from the sample. Cold antigen, amount of cold antigen is inversely proportional to radioactivity of the sample. So more is the cold antigen presence, less is the radioactivity, because more is the cold antigen, which means that more amount of cold antigen has replaced the hot antigens, and hence the radioactivity will be decreased. So keeping this in mind, we do our calculation and we compare the graph with the standard curve to accurately obtain the concentration of cold antigen in a patient's sample. So let's now discuss about a few of the applications of radioimmunoassay. So one main application is used in the cancer detection. It is also used in hormone level measurement. This all are basic applications. Hormone level measurement, and it is also used to study drug efficacy. Alright, and then we can also screen the blood for HIV and other positive pathogens, right? And these are few examples of radioimmunoassay, and that's my topic. Thank you.